Welcome all of you. In the last class, I introduced you to the concept of allelochemicals and I concluded the class with the classification of 14 different groups of allelochemicals which are known today. And one of the aspect what I highlighted in the last class is eventually any of these success or a failure of a invasive species and failure of a crop or a specific life form is because of some specific chemical reasons. And today we will be exploring some of those chemical reasons which leads to the invasion of one species over the other one, because that is the way how we could develop strategies and sensory devices to counter this menace of invasion. So, getting back to the slides, if you look at this slide where we have shown the global spread of Varvacina enceloides, there is one interesting thing you will observe, that if it is starting from somewhere out here and it is spreading all over the world. Now, you might wonder how such small number of seeds are spreading all over the world, because say for example, if you introduce some x number of seeds from a region A to region B by some means, either it is a animal introduction, human interaction, whichever way it got introduced into the new region, how come those x number of seeds compete with such a large population? Because when you are invading, you cannot take a huge army, it is very small army which goes there and it invades. How that happens? And that is one of the very fundamental question which has been boggling the evolutionary biologists who are working on theories of invasion. So, in that context, in the last class I highlighted, we will be talking about a novel weapon hypothesis. So, from here I once again highlight what are shown in red, the allelopathic interference, which I have already talked, and that supports the novel weapon hypothesis. This is based on Dr. Batish's earlier work. Now, in terms of the chemicals, what I have described in the last class, you could see there are 14 group of chemicals which are present, and specifically I put a box in one of them which says that guanine derivatives, these are the ones which we will be discussing further. Now, what really is novel weapon hypothesis? So, I have to give you an example of it before I show you it on the slide. Say for example, one species is moving to another region. Now, once again, it can move through animals, through insect, through natural processes, through human, or it has been purposefully introduced. Now, what are the options it has? One option is that it competes over a period of time, say 10 years, 20 years, 50 years, 100 years, and eventually establish itself. Or it perishes, it fails to invade into that region. Or it may develop some different kind of a strategy by which it can outsmart the existing large population. So, in a way, you can visualize a small army invading a huge country by using some very sophisticated instrumentation, which the country which has been invaded does not possess. So, in other words, that small army of invaders possesses some extraordinarily well developed sophisticated weaponry. Now, that brings us to some of the historical things. If you look at the slide, There is a very interesting slide I put across, and the reference has been given there, where it has been shown when during the 13th century in the Mongols, rather 12th and 13th century, when the Mongols were invading across Europe, one of the reasons why they were very successful, though Mongols remain extremely disciplined and extraordinarily great fighters, but along with that, they possessed something what they called as the recurve bow carried by most of the invaders. This recurve bow at that time was something which no one ever heard before, and that gave them some extraordinary advantage. So, if you look at it, what it did actually, the recurve bow fired arrows faster, further and with greater force than anything the Europeans had ever experienced. Such novel weapons have played a role in many human invasions, but novel biological weapons have only recently been considered possible drivers of invasion by non-human exotic species. So, this is the catch. So, this is another emerging hypothesis in evolutionary biology, what says that if you possess a novel weapon, but when we talk about a novel weapon, then you have to understand what is that novel weapon. That brings us to the novel weapon in the chemical ecology of nature. So, as I mentioned just few minutes back, one of the mysteries in ecology is how exotic plants occurring at low densities in their native ranges attain incredibly high densities in their introduced ranges. That is exactly what I was trying to tell you while showing you this map. How come those a small number of species could spread all across the world and completely invaded and penetrated into those ecosystems. So, in that regard, there are, as I mentioned, there are multiple hypotheses. One is the natural enemy hypothesis. These are natural enemies of those plants existing in another place. But how that could happen, because you belong to one geographic biogeographic region, how could you have enemies in another biogeographic region? Then there is another concept called the evolution of increased competitive ability. So, they have a better competitive ability, but all these things, when we talk about competitive ability, natural enemy hypothesis, you have to have certain physical means to do so. You should have some chemoevolutionary advantage, some kind of chemical cue has to be there, otherwise it is extremely difficult to justify these kind of statements. And that is exactly where this course will always try to inspire you. Look for the chemical signature, look for that molecule, look for that signature which justifies this hypothesis. And the third one what we talk about is the novel weapon hypothesis, or allelopathic advantage against resident species, AARS. Now, this is interesting. Say for example, one particular plant which is an invasive plant moves to a newer biogeographic region and it secretes certain kind of allelochemicals which the native plant is not used to, and that allelochemical could act as an inhibitor of growth or could lead to some form of inflammatory reaction onto the host plants. And by virtue of that, this newer plant can stunt the growth and or may lead to the death of the existing plants in that surrounding. So, in nutshell, if you talk about the novel weapon hypothesis, so it says something like this: the success of some exotic invasive plant species may be due to possession of novel weapon, or in other words biochemicals that native species have never encountered. So, they are not at all aware of it. The second thing, the novel weapon hypothesis raises the possibility of coevolution among plants in different regions of earth, and that, listen to this carefully, mixing species from different regions. So, you are introducing something from one region to another region, increases the chances of disrupting the ecological balance or ecological processes that led to species coexistence and greater community diversity. Third and important part, novel weapons suggest an alternative mechanism for evolution of increased competitive ability in invasive plants. If invader possesses allochemical weapons that provide greater competitive advantage in their new habitat than in their original ranges, then selection may act directly on those traits, and this is called allelopathic advantage against resident species hypothesis, or AARS hypothesis. So, now we will revisit Professor Daisy Rani Batish's earlier work on Verbacina insuloides, where in one of her beautiful paper, she highlighted how this particular weed could spread all across the Indian subcontinent as well as in Australia, and as far as it could spread its reach, because her group discovered that these plants secrete, if you follow these lines which are mentioned in red out here, however, a guanidine derivative, galagine, was only found in the fresh leaf materials of the plant. The findings support the novel weapon hypothesis and suggest that Verbacina competitively exclude its neighboring plants by virtue of allelopathic interference. So, you realize that there is one single compound from the armory. So, now I will take you back to that specific slide where I said that I put a box on this, specifically this particular guanidine derivative, because this is the one which is a matter of our concern. Okay. So, now you see, if we have to have a sensor, biochemical sensor or biosensor, whichever you call it, or a chemical sensor, we should know the nature of these molecules and we should have a way to detect them, and if you want to control, we should have a way to nullify them by some other means. How we do so, we will talk about it. But having said this, I will move to slightly different aspect before getting back to this allochemical concept. If you look at this slide now, the economic burden of the which are underestimated. So, one of the work which was conducted by Dr. Alok Bang from evolutionary biologist from Azim Premji University, what he co-authored in 2022 showed that invasive alien species, as we talked about, cost India $127.3 billion in the last 60 years. As a matter of fact, making India the most invasion cost bearing country after United States. So, think of it, the loss reflect the damage from only 10 out of 330 known invasive species. So, we have no idea about another 320 species, how they are causing damage. This cost estimation is like mind boggling. And if you look at the globally, there are 37,000 established alien species cost the economy over $423 billion annually, negatively impacting human health, biodiversity and food system. As simple as something like this. Say for example, you have mustard crops growing and there is an invasive crop which has very similar seeds like mustard, they get mixed into the oil seeds. Sometime human being introduce them. So, what happens is that the quality of oil is compromised and oil may come with certain chemicals which are not favorable for human health or human consumptions. This is one of the most simplest example I am giving you. So, if we look at the slide, over 3,500 of these invasive alien species severely harming biodiversity and human livelihood. The costs have quadrupled every decade since 1970, and the Intergovernment Society Policy Platform on Biodiversity Ecosystem Services has been highlighting this over the years through the decades. Okay? And if we look at specifically the case of India, India is among the countries that does not, or they don't, despite specific targets related to IS mandated in the National Biodiversity Action Plan. So, in a way, so far we do not have a very strong platform, legal platform I must say, to prevent or to stop the menace of these invasive alien species of plants, possibly microbes and other organisms which are creating a lot of fluctuations in our economy and mostly having detrimental effect in our economy. So, these are some of the points for you guys to ponder why we really need to understand the fundamental concept of biosecurity, because these are those points which we kind of overlook when you look at the productivity of crops, productivity of other agro products. Okay. Now, from here I move to a opposite concept, again back to the allelochemicals. So, when we talk about novel weapon hypothesis, we said that a alien species is having a negative impact on the native species by virtue of secretion of some unique kind of chemical. But does native species produce allelochemicals? Interestingly, allelochemical production is not a proprietary of weeds or alien species. Every plant produces allelochemicals. Now, things that do we understand? So, say for example, I will give you a situation. If you look at rice, momelactone A and B, resorcinols, cyclohexanone, flavones, glycoside derivatives, and there are several secondary plant metabolites such as phenols, coumarins, terpenoids, steroids. To minimize losses, these are all identified allelochemicals. Now, to minimize losses, 3 million tons of chemical herbicides are currently used annually, or synthetic herbicides which are used annually worldwide to control paddy weeds in agriculture system. Now, think of a system, just as I mentioned, think opposite. If rice has the ability to produce these allelochemicals, what are the chances that we could use those allelochemicals as herbicides? It's just I am asking you to think the reverse way. So far I said the allelochemicals are those novel tools or novel weapons by which a alien species invade a native population. Now, native population also produces its own repertoire of allelochemicals. Now, what we do is that they have their own natural defense mechanism. Now, one thing I will highlight here, which is slightly digressing from the main frame. If we look at the history of herbicides, weedicides, pesticides, this is the history of 19th century. It is pretty much after 1930s and 40s or during 1940s, humankind started using these kind of chemicals for crop production. Prior to that, there was no such synthetic chemical compounds which were being used at such a large scale. The point comes how the plant used to defend itself. Weeds were still there. The weeds were there since time immemorial. Now, one of the things could be a plant which is searching for its nutrients in a normal scenario of say 100 years back when there were no fertilizers. It developed a better immune profile. In other words, those rice plant, wheat plants used to produce more allelochemicals in order to fight with the wheat crops growing around them. But with Green Revolution, with the discovery of Haber-Bosch process, followed by the a readily available nitrogen fertilizer, potash, phosphorus. Most of these plants were getting their food pretty readily in the soil and in the upper part of the soil. So, their fighting capacity get compromised. So, in other words, what if these plants, these rice, wheat, which are growing post green revolution, have a compromised ability to produce allelochemicals, so that their ability to fight with the weeds got reduced because they were not exposed to tough situation. They were never exposed to those kind of stress which these plants were experiencing prior to green revolution, prior to the discovery of chemically synthesized herbicides, weedicides and pesticides. This is the thought which always runs through my mind, that what if that is still those wild varieties of say rice, millet, sorghum, they possess a higher level of allelochemicals if grown in those hardy situation without giving them fertilizer, versus those crops which have been fed ad libitum of fertilizer. Because when I was looking through this particular set of data of rice, this particular idea crisscrossed my mind, that if rice is producing this much and if you follow it, wheat produces the phenolic acid, hydroxamic acid and fatty acids. So, it means each one of these plants have a natural ability to produce these kind of herbicides or these kind of allelochemicals. Now, those allelochemicals are in a way plants' natural herbicidal or weedicidal properties. Okay. Now, what we have done in last 1000 years, which are non-chemical alternative, is we followed something, the concept of intercropping. Intercropping has been done in such a way, say for example, there is a plant A and a plant B. So, the plant B secretes something which prevents the growth of weeds and thereby promoting the growth of plant A, or vice versa, plant A secretes certain things which prevent the weeds to grow and allow a better growth of plant B. Similarly, crop rotation, you rotate the crop, this season you have rice, next season you have some short term crop, then you move to wheat, then again you move to some other short term crop, so on and so forth. That way you are completely changing the microbial niche of the soil. In a way, by doing so, you are not allowing the weed or the invasive species to settle down, unless they are so invasive that they could adapt to any situation. In other words, you are continuously changing the microbiota and the milieu of the soil. Next thing is mulching, where you cover the soil surface and prevent something to grow. So, these are the strategies which have been done over the years to prevent weeds to grow. Now, what I discussed just prior to this, if we have a exactly opposite strategy, say for example, we know that these are the chemicals which are causing damage to the host plant or to the native plant, and we know the native plant has the ability to withstand it. So, if we could extract those allelochemicals and purify them, and then what we do, we identify them using different kind of mass spectroscopy and other techniques, and then we see the impact of these allelochemicals on the different plants, and then we go for industrial processing. So, this is how we can utilize one of the very basic chemical tool of nature itself to develop bioherbicides. Similarly, even those invasive allelochemicals could be used as bioherbicides in places where there are other weeds which are destroyed that ecosystem. So, you see that things which may have detrimental effect could also be used for positive purpose. So, it is up to us how we tweak the chemical nature, but in order to understand that, we have to think the way nature fights its own survival strategies, or it develops its own survival strategies. So, if we understand that we will be able to have sustainable design strategies for bioherbicides. So, there are two concepts what we followed. We talked about the novel weapon hypothesis, then we talked about that chemical nature of these novel weapons, which in terms of allelopathic chemicals could be classified into 14 different groups. Then we talk about a case study of Professor Daisy Rani Batish's work on one specific wheat and the guanine derivative what it produces, which is unique novel weapon. Next, we talked about the ability of all these crop plants, whether it is rice, wheat, sorghum, millet, the allelochemicals they produce and how we could capitalize on them, and we can capitalize on the allelochemicals produced by the invasive species in order to develop bioherbicides which degrades in nature and saves the soil from detrimental effect. So, that is where we are closing this class. Thank you.